Intro. I have spent a lifetime watching the silent, subtle dramas of the natural world. Few senses intrigue me more than a shark's capacity to perceive chemicals in the sea. I'll myth-bust common beliefs about how sharks detect blood, separating wonder from exaggeration. Can sharks smell a single drop of blood? Let's find out. I promise 10 science-backed tips and clear explanations, grounded in laboratory work, field observation, comparative anatomy. We'll explore olfactory organs, fluid dynamics, behavior, and other senses. We'll also reveal limits set by physics and environment. Stay with me. We'll move from the anatomy of the nose to the movements of the ocean, from elegant experiment to gritty reality. By the end, you'll understand how a shark locates and homes in on distant chemical cues without resorting to myth. Chapter 1. I have often heard the dramatic claim that a single drop of blood can lure an ocean of teeth, and it is true that many sharks can detect remarkably faint chemical traces. Scientific reports describe some species detecting concentrations as low as 1 drop of blood diluted into millions of drops of seawater, but that statement requires careful context. Detection thresholds measured in controlled settings do not translate directly to open ocean encounters, where currents and turbulence play a dominant role. When we speak of an extremely powerful sense of smell, we mean that the sensory machinery is highly sensitive and tuned to particular molecules, especially amino acids and other compounds associated with damaged tissue. Sensitivity varies widely between species. Some deepwater and scavenging sharks are tuned differently from coastal predators that hunt using sight as well as smell. The phrase powerful therefore reflects both biological specialization and ecological need. It is also important to separate detection from localization. A shark might detect a faint chemical cue at low concentration, but still be unable to determine where it originated without further sampling and comparison. The ocean is a dynamic medium, so the initial sniff must be followed by active behavior, turns, pauses, zigzags to convert a chemical whisper into a clear directional signal. Chapter 2. Much of a shark's brain is devoted to processing smell. The olfactory bulbs are relatively large and richly supplied with nerve endings. I have watched specimens in laboratory settings where a flood of distinct chemical cues produces immediate and measurable neural responses, demonstrating how rapidly the olfactory lobes analyze complex molecular information. These bulbs do not merely detect, they begin the work of classification, separating food-related signals from background chemistry. Anatomically, the olfactory sacs in sharks are proportionally bigger than in many other fishes, reflecting the evolutionary premium placed on smell. The neural pathways that lead from the nostrils to the brain are direct and efficient, enabling a swift behavioral response when a relevant scent is encountered. In predatory species, the olfactory centers are integrated with motor circuits so that smell can precipitate approach behavior. This organization means the brain is prepared to interpret cues in ecological terms: potential food, a mate, or merely familiar seawater. Chapter 3. A subtle but crucial feature of shark noses. They have 2 separate nostrils, each sampling independently from left and right. I find this bilateral sampling remarkable. It functions much like stereo hearing, vision allowing the animal to compare concentrations across a very small spatial baseline. When scent concentrations differ between nostrils, the shark can infer a direction and adjust its heading accordingly. This comparison is not instantaneous like a radar ping. Rather, it is part of a dynamic process. A shark will often swim with its head angled, perform gentle turns, so each nostril receives a slightly different water parcel. The brain compares those signals, guides corrective movements, a dance of sampling steering that gradually narrows the search area. Chapter 4. Laboratory tests have been instrumental in quantifying what sharks can actually detect, and the results are impressive, without being mystical. Some species can respond to chemical concentrations on the order of parts per million or even lower in carefully controlled conditions. I have observed experiments where tiny puffs of amino acid solutions elicit measurable changes in swimming and orientation. Chapter 5. Detecting a chemical is merely the opening act. The real skill lies in extracting direction from a variable, turbulent plume. Sharks do not blindly charge en masse toward a scent. Instead, they adopt a zigzagging, Boustrophodon-like search pattern, sampling across the width of a plume to find where the concentration is strongest. I have seen footage in which a shark alternates left-right turns, each pivot refining its estimate of the source direction. This behavior resembles strategies used by many animals that rely on olfaction: counter-turns, upwind movement, bursts of straight swimming followed by resampling. In water, this is adjusted to current flow rather than wind, so the shark often orients upstream of the scent to encounter fresher, less diluted water. The pattern is not random but a systematic method for converting noisy chemical information into a navigational cue. When turbulence is high and the plume fragments into filaments, the shark will shorten its sampling excursions and rely more on close-range senses. When the plume is coherent and steady, it can make longer, more decisive moves toward the source. Chapter 6. Sharks do not simply inhale seawater like a terrestrial animal. They rely on a continual flow of water through their nostrils as they move. This swimming-induced flow ensures fresh samples are constantly presented, providing a temporal sequence the brain can analyze. Olfaction often initiates an investigation, but smell rarely acts alone. Sharks rapidly integrate vision, hearing, touch to confirm and refine the information they receive. I recall observing a shark that detected a chemical cue and then switched to visually scanning the area, narrowing its approach as sight provided spatial detail. Sound and pressure cues can also indicate struggling prey, while the lateral line and ampullae of Lorenzini supply close-range confirmation. This multisensory integration is both efficient and economical. A faint scent might trigger a cautious approach, but only when visual or mechanosensory confirmation occurs does the shark commit to an attack. Once a shark closes the distance to potential prey, an additional and extraordinary sense comes into play: electroreception, via the ampullae of Lorenzini. These tiny jelly-filled pores detect minute bioelectric fields generated by muscle contractions generated by nerve activity in living organisms. In the final meters, a shark shifts from olfactory cues to visual cues and then to electrical whispers. Electroreception localizes prey hidden under sand and works in low light, giving precise spatial information. The lateral line is another close-range instrument, detecting the vibrations and pressure gradients created by moving or struggling animals. When a creature thrashes, it generates a cascade of mechanical signals that the lateral line transduces into directional information. I have seen footage where the moment a prey item begins to struggle, the shark homes in almost immediately, guided by these hydrodynamic cues. The lateral line and olfactory system thus work in concert. Chemical detection brings the predator close. Mechanical signals confirm and locate the prey precisely. The lateral line is effective where vision is poor and where chemical gradients are noisy. Vibrations propagate through water in ways less susceptible to turbulent mixing. For many shark species, the signature of motion can be the decisive factor triggering a rapid and accurate strike. Finally, and perhaps most importantly for myth-busting, the environment governs how chemical cues behave. Currents, temperature gradients, turbulence, salinity all influence dispersion. Thus, the same single drop of blood under calm, steady current might travel as a coherent plume, whereas in rough seas, it dissipates into undetectable fragments. This variability makes absolute statements about detection range misleading. Sharks are superb chemical detectors, but detection depends on anatomy and environment, not supernatural noses. I hope this exploration has replaced sensational myth with careful, awe-filled understanding. The combination of large olfactory organs, bilateral sampling, active search behavior, complementary senses produces a predator exquisitely adapted to its world. We have seen how lab measurements and field observations combine to give a full picture. Environmental dynamics ultimately shape what a shark can detect and how it acts. If you enjoyed this myth-busting dive, like, subscribe, and tell us which shark fact surprised you. Thank you for joining me and Hush Luxury on this journey beneath the waves.